questions now that you had a chance to process things? So over the break, I was trying to find another YouTube video that I wanted to show, and I didn't find what I wanted to. Actually, I got sort of sidetracked because when you do a search on YouTube, it'll have a little indication when you've watched videos. And this came up in a search that I was trying to do, and it didn't indicate that I ever watched it, which suggests to me that it might be a new video since I last looked through for these things. So I was sort of previewing it a little bit during the break, and I think it might be a pretty good video. And it touches on a few things that I would like to include in what I was saying. And I think it'll be hopefully a kind of nice review of sort of the whole idea of what I did in the first part. So let me go ahead and play this real quick.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of the membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Okay. So, oh, actually, I didn't want to close that out yet. I want to kind of go back and touch on a few things. One, I kind of like how in this, it's sort of trying to set things up similar to the way that I did, just showing you some cells within a circuit controlling this. And this is a much more simple circuit than what I'm talking about with the shower and all that. This is a very simple reflex, just heat and pain stimuli will go to the spinal cord and immediately cause contraction of the biceps brachii to withdraw the hand. What I presented with the shower temperature thing, I wanted to include more than just a simple reflex. So I did want to include the information going up to the conscious regions of the brain that would cause a voluntary change in movement. But the point here is fine. And they do a nice job of kind of illustrating the parts there. They introduced the name or the word interneuron here, which I don't tend to use too much. It just means a neuron that's between two other neurons. So we have three neurons represented here. This is a sensory neuron, meaning that its dendrites are receiving sensory stimuli, and a motor neuron, meaning that its axon causes contraction of the muscle. And then the inner neuron in between is just receiving input from the sensory neuron, and it's sending its output to the motor neuron. So in this sense, inner neuron just means a neuron in between two other neurons. I'm not crazy about that distinction because in what I was showing you, there were three neurons that bring the sensory information up to the brain. Those technically should all be considered sensory neurons because they're carrying sensory information. But the second and third one could also be considered inner neurons. But I think it takes away a little bit of the functional aspect of what's going on in the greater circuit. So let's see, what else do I want to point out? Oh, the whole time when it's animating the membrane and talking about what's going on, this thing is always working. This is the sodium potassium ATPase. It hydrolyzes ATP to power pumping 3 sodium ions out of the cell and 2 potassium ions into the cell. Okay, so that's one cycle of it. So 3 sodium ions come in, it hydrolyzes ATP, and the 3 sodium ions are pumped out. And then 2 potassiums come in, and they're pumped inside, and then it just repeats that thing over. What's kind of nice about this is throughout the whole sequence where they're showing the membrane up close, that's always going on, which is a good representation. That often gets left out. The sodium potassium ATPase is always pumping those ions in and out of the cell against their concentration gradients to keep the concentration up. Okay, keep the concentration gradients up. And then let's see. as it's going through this stuff, up in the corner, it shows that graph, just like what I showed. And when it gets down past the hyperpolarization and it returns to its resting potential, it's explaining that the sodium-potassium ATPase is playing a role in getting things back the way it should be, which is a good representation. Now, one thing that's a little unfortunate, I suppose, is here we have for purple the, I think that's how she did it. Purple is the potassium channel and green is the sodium channel. Yeah, because that's the colors that correspond there. They're drawn in this to look pretty much the same, not identical, but they kind of work on seemingly the same principle. But actually, as I was describing the sodium channel, it actually has those two gates. And that's not really represented here. Okay. There's a lot of good things in this. So the fact that they left that out is fine. Sure.
Okay, so I understand that the blue one's pumping it in and pumping it out.
This one,
But
yeah.
when it was playing, there was the green stuff coming out of the green channel coming back in here while the blue one's pumping it out. That's normal.
Right. So this is always doing its job, always going on. When the stimulus gets it up to threshold, then the green channel will open and sodium's being pumped out all the time. But once this channel opens up, it rushes in. Okay. And as soon as it rushes in, it's going to start being pumped out again. Okay. But the rate at which it comes in is going to change the membrane potential and get us up here. So that, yeah, that is, those two things are going on at the same time. And that's actually kind of what I was saying. I like seeing this always happening when they're looking here, because that is, it's true. That's always happening. But it helps to keep the sodium outside and the potassium inside. It keeps doing its job, but really when these channels are opening, what it's doing is kind of lost in the background. Now, when it was showing this part, it actually made an interesting point, kind of in the way that it was, well, in the voiceover and in the way it's animated. The ions aren't moving very far. So there's sodium here, and it moves in here. There's potassium here, and it moves out there. It's not really huge movements of ions across. They're all kind of right at the membrane, waiting to move across. So all of the electrical changes we see here happen very quickly. Okay, so that's an interesting point that they made there. And then it gets into talking about the synapse, which we've talked a little bit about, and we're going to come back to and talk more about here. So it's kind of nice that you had a little review there. What I was actually looking for as I came across this was I was trying to find a video about, whoops. Here we go. I guess I just didn't finish typing all that in and I found that video. But I was trying to look up action potential propagation because, so here's where in the book I'm talking about the action potential. The next thing is propagation. Okay. And the reason I was looking that up on YouTube is that there used to be a great video of that, and I'm not seeing it. So there are a number here that I've watched, and I don't know if there's any that are that great. This one here, let me play this. It's only about a minute long.
Depicted at an action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
Okay. So, bless you. What that's getting at, which is sort of the beginning of what this is saying, is this is a depiction of the action potential measured by time, so the voltage changes by time. We can also consider this in terms of what's happening on the membrane by location. Now, if we're measuring that, we'd actually have to turn this around, and we'd go from the cell body to the end of the axon here, and we'd see the membranes at rest, and then something changes it to get to threshold, and it depolarizes and repolarizes and hyperpolarizes. Okay. This series of events moves down the axon from the cell body to the end of the axon, which was what this was kind of depicting. Okay. What's going on in this little red band here is the peak of the action potential when it's positive inside compared to outside. And that's going to keep moving down the axon. Okay, so here, the red band's moving along the length because we could sort of put that peak of the action potential right there, and it just keeps moving along there. Behind the action potential, so the area right here, closer to the cell body, is its membrane potential is going to look like this part of the curve. At this point here, the axon potential can't be restarted. Okay, the axon, is in what's called the refractory period, which basically means another action potential cannot be started. And there's actually two phases of the refractory period. There's the absolute refractory period, which is absolutely no action potential, capitalized, abbreviated AP, can be started. And this is because, oh, what did I do? Oh, I did it again. What is going on here? There we go. This is because the sodium channel is inactivated. Remember, there's an activation gate and an inactivation gate. While that inactivation gate is closed, we call the channel inactivated. And no sodium is going to get through, and nothing's going to open it back up until it gets back to normal. Okay. The other one is the relative refractory period, which is a new action potential requires a stronger stimulus. Okay. And this is because potassium is leaving the cell. So what this is getting at is, oh, let me just talk about this and I'll put it back up on the screen. When the sodium channel is open during depolarization, we can't open it more. So we're going to see depolarization. But once it inactivates, and then we're in the repolarization part, we can't reopen that sodium channel. So we're in that absolute refractory period, okay? And we're going to repolarize. Somewhere down here, probably time-wise, the sodium channel's back to its normal state, and we could reopen it. But if we reopen it, the sodium that might come in has to combat the flow of potassium out. So while 15 millivolts of depolarization got us from -70 to -55 here, 15 millivolts of depolarization coming in during the relative refractory period is just going to soften the hyperpolarization. Okay, it's not going to get it back up to threshold. We could start here. Okay, if this is when the relative refractory period starts, opening sodium channels at this point is not going to turn the curve around immediately to go back up here. It has to slow down and then get back up here. Okay, so this much of a stimulus, 15 millivolts, will slow this down but not get it up to threshold. You have to have a really strong stimulus to slow it down and get it up to threshold. Okay, so a stronger stimulus has to come along. It's not until hyperpolarization is over and we're back to resting potential that the same stimulus can fire it off. Okay. As we're moving along the axon, behind the action potential, right here, it's in the absolute refractory period, and it's in the relative refractory period here, and it's back to rest here. Okay. So as they're showing the current of charge where the it's positive here, the positive is going to move over actually in both directions. It'll get to threshold on this side, but it won't get the threshold on this side. So then action potential will move here, and then the action potential will move there, and the action potential will move down like that. Okay, that's what propagation is. Okay. The word propagation is related to the word propaganda. Okay. Propaganda is usually means the spreading of false information. But really, it's called propaganda when you don't agree with it. It's the truth when you agree with it. But the idea behind propaganda is the spreading of information. Okay? And so the propagation of the action potential is the spreading of the action potential. And because of the refractory period, it only spreads in one direction. Okay? The sodium enters into the cell and will actually diffuse both directions. But when it diffuses back to where the action potential just came from, it can't depolarize to threshold again because of the refractory. So it can only go in one direction, and it propagates down the length of the axon. Okay. Now the video I was hoping to find is talking about propagation in two different types of axons. There are myelinated axons and unmyelinated axons. And so that little video I just showed you there is an unmyelinated axon. Sodium rushes in through the membrane where the action potential is occurring, and it spreads to either side, and it propagates in one direction. And more sodium rushes in right next to that. That propagation will travel at probably about, I think it's 2 meters per second or something like that, which is fairly slow for an electrical signal. In a myelinated axon, it works a little bit differently, and the speed of propagation can be much higher. It can be 50 to 100 meters per second. And the reason that that works is because of what the myelin is doing. Okay. Let me just go back into the nervous tissue section and pull up this picture of the neuron that we were looking at before. Okay. So here's myelin wrapped around part of the axon. The propagation we just looked at would be sodium rushes in here and spreads, and more sodium rushes in here and spreads, and more sodium rushes in here and spreads, more sodium rushes in here and spreads. Where the myelin is, sodium will rush in here and it spreads, and it has to keep spreading. It doesn't have anywhere to go until it gets to the first node of Ranvier. And then at this point, there are new sodium channels that can open, and sodium will rush in here, and then it can spread down this way. And then sodium can rush in here, and it can spread down this way. And then come in here, spread down this way. Now, the spreading part happens pretty quickly. It's the opening of the channels and the moving of ions across the membrane that slows it down. So without myelin, it moves kind of slowly along here. But once it hits the myelin, it goes here, then it goes here, then it goes here, and then it goes here, it goes here. And it jumps from node to node. The distance between nodes is actually the optimal distance for the spreading of the ions in the axon. So they're just sort of showing, for the sake of animation, 3 plus signs here. But the positive ions, positive sodium ions, will spread down the length of the axon and diffuse within the space here, and some of it might leak out. The length of the myelination, say from here to here, is exactly how the length of the axon that can sustain the depolarization at the level of threshold. Okay? So as it's entering in here, the action potential is taking place, it depolarizes it to positive 30 millivolts. As that sodium spreads down here towards the next node of Ranvier, that distance is precisely how far the sodium can move to still reach threshold when it gets there. Okay? And once we get there and reach threshold, more sodium rushes in, we get positive 30 millivolt peak, and the sodium rushes this way. Okay? I say that it's precisely the right distance because it's something that the system has adapted to. Myelinated axons are found in all sorts of animals. And so it's something that's been around for a very long time. Sometime way back in the mists of time, when myelin was first sort of coming to be prevalent, there was some variation in how long the myelin sheath was, the distance between nodes. And that variation was probably based on some gene that established how big the oligodendrocyte or swan cell got. When it was really short, let's pretend it started with this actual length. When it was really short, it helped speed up the action potential a little bit, but maybe not a whole lot. And as it started getting longer and longer, the speed of the action potential got quicker and quicker. Okay? Eventually, it gets to a point where it's too long, and the sodium coming in, spreading down to the next node, diffuses so much that the membrane is no longer depolarized enough to open the channels. So imagine 3 different organisms. Okay? One organism has short myelin sheaths. Another organism has overly long myelin sheaths. And then the third organism has perfect length or optimal length myelin sheaths. Okay? And all 3 organisms are of the same species. If they survive, they can pass their genetics on to offspring, and they'll have the same kind of distance between their myelin sheaths. Now, the organism with the overly long myelin sheaths will not be able to propagate action potentials along its axons at all. Okay? It'll get started and it'll peter out before it gets to the end. The one with the short myelin sheaths can propagate action potentials, but they're slower compared to the one with the optimal length. Okay. So you have these 3 organisms chewing or feasting on grass in the savannah or something like that. and a predator comes along, okay? The ears, and for some reason I imagine these are bunny rabbits, but whatever. The ears hear the sound, okay? And that sound is translated into an electrical impulse through gated, I think it's a mechanically gated channel. The action potential then travels along the nerve from the ear to the brain to let the organism know that there's something out there. So all three of these organisms hear the predator. The one with the optimal length myelin sheaths hears it first and can react fastest. The one with the short myelin sheaths hears it second, can react, but a little bit slower than the first one. The one with the overly long ones never hears it because the signal never gets transmitted from the ear to the brain. Okay? So we know the fate of that guy. What's going to happen to him? Going to get eaten. So we get rid of the first guy. He's not going to pass his genes on because the myelin's too long. Okay. So we have the other two. The optimal length myelin will hear it first and react first and be able to get away. Okay. The one with the shorter myelin hears it a little bit later, microseconds later maybe, but still a little bit later. And it's going to react second. Okay. And then the reaction is going to be sending signals to muscles to contract so it can run away. The organism with the optimal length myelin is going to send the signals to the muscles quicker, and they're going to contract and it's going to run away quicker. The one with the short myelin sheaths will send signals to the muscles, but they'll get the signal a little bit later, and they won't contract as quickly. There won't be continuing signals coming to it to maintain the contraction for running. So while it might only be microseconds in difference between when the two respond to the signal, the one with the optimal length myelin is going to run faster. Okay? It's going to be able to control its muscles more efficiently and get away quicker. And so the one with the shorter ones won't be able to get away as quickly. And if the predator catches up with him, he's food also. And so the one that's left over that was able to get away is the one with the optimal length myelin sheaths. And that gets passed on. Okay? Now, I'm simplifying the idea a little bit, talking about three organisms, but over all of the organisms where this trait got optimized, it's going to reach the perfect length from one node to the next to get the best transmission times, best propagation. And so by the time we get to humans, we're no longer adapting that aspect of our nervous system. It's where it's supposed to be for us. And it's probably been that way for eons, and it was originally established in some organism that is long gone and mostly forgotten. But the distance between these is really optimal to get the best propagation speed. So like I said, without myelin, which is what this video is actually showing us, propagation is going to be fairly slow, maybe 2 meters per second. But if we can myelinate these axons, the speed of propagation goes up quite a bit to maybe 50 meters per second. Yeah.
So if someone has a delayed response to touching something hot,
Yeah.
that has to do with the myelin?
It can. That's not going to be the only reason. But some people with demyelinating diseases like multiple sclerosis or ALS or something like that, I think those are both demyelinating, they might have that kind of problem, that the signals don't get into the system quick enough. Now, we are kind of talking about small changes in speed. So if somebody touches a hot stove and there's a myelination problem and the signal's not traveling in fast enough, it might be a difference in milliseconds or hundreds of milliseconds, but it can be enough to cause more tissue damage. So it's a fraction of a second, but a fraction of a second might be enough to make the difference between a first degree and a second degree burn or something like that. So yeah, it's possible, but there are other factors that could play a role in that.
Like as you get older, your response could be
Certainly,
lower.
the response times do decrease with age, which I suppose you could investigate just by looking at driving. Okay, as people get older, their ability to respond to things in the environment around them will be decreased. That's not necessarily a matter of myelination or propagation speed. It's more in the brain, connections change a little bit with age, and the signal to noise ratio gets worse. And so it's not conscious distractors. It's just neurons have too much information to make sense of, and they don't respond to it that quickly sometimes. Actually, age-related decline in cognitive function is sort of related to what I did my dissertation on. So that's where I would kind of go with that. But there might be other reasons for that also. So I pulled this one up because it's kind of short. It's only about a minute. It did give you a good idea of how propagation works within a unmyelinated axon. And then over here, here's a slightly longer video of how it's propagated. And this very well might include myelination. And then this one here says myelin and saltatory conduction. It's much longer, and I can see from the little thumbnail that it's not an animation like this so much as a series of drawings. Saltatory conduction is what we call that type of propagation that goes from node to node to node to node to node, jumping along. I really wish I could find that other video because it's a really good representation of that. They must have taken it down. I don't know where it went, but there's a really good video that shows actually they have an unmyelinated axon and a myelinated axon both in the same frame, and you can sort of see a difference in the speed of propagation there. But the video I want to show you, it's not on YouTube anymore. So, so where were we? Back to this. So that was the action potential stuff. And after explaining the action potential, the next part is about propagation. I don't have a picture to go along with that. The video that I just showed you is pretty good representation of that idea. Then the next section is called communication between neurons. And this is really getting at, well, it's going to talk a little bit more about obviously graded potentials, but what it's getting at is what's happening in the synapse. So this figure here is getting at a little bit about graded potentials like I was talking about before. And what this is showing is graded potentials can be different sizes. So this one goes up to maybe -64 millivolts. This one goes up to -60. This one goes up to -61. Okay. This one goes up to -52 or something like that. The size of these different things has to do with the size, the strength of the stimulus. Okay. Another thing in here, this one is wider than these two because the stimulus can last longer too. And that'll make a difference in the shape of the graded potential. Now, of these four, the only one that's strong enough to actually get to threshold is this one right here. Okay, just to show you that there is some variation there. They don't have to be depolarizing potentials. There are other types of potentials where the membrane gets more negative. That's what we call hyperpolarizing. And so again, they can be different sizes because they're graded potentials. So we can see a slight hyperpolarization, a greater hyperpolarization, or a huge hyperpolarization there. Okay. So this is just setting up sort of what different types of graded potentials can be. The thing is that graded potentials can be added together. Okay. Summation, adding together, means that we can take these separate things, and if they occur together, they add up. So if we have one that depolarizes from -70 to -64, and another one that depolarizes from -70 to -60, that means this is a a 6 millivolt depolarization. This is a 10 millivolt depolarization. They happen at the same time, they'll add up to a 16 millivolt depolarization. And so we can, by summating, we can get to threshold even with seemingly weak stimuli that are giving us these potentials here. And so by adding together graded potentials, we can get the threshold. We can also summate in hyperpolarizing potentials. And the point here is it moves us farther away from threshold. So if we add this, say, 4 millivolt hyperpolarization to this 6 millivolt depolarization, the end result is going to be 2 millivolts of depolarization. Okay, they kind of cancel each other out in a weighted way. Yeah.
So if the stimulus doesn't reach threshold and like an action potential doesn't occur, does the body not respond to that stimulus, or?
Right. So that's kind of hard to give you a sort of working example of, but let me try. So as I was talking about the types of things that can be stimuli, all of those different stimuli that I mentioned, and like I whispered and yelled. Can I help you?
Our teacher said to put the projects in here.
Who's your teacher?
Right here.
Okay, I'll have to talk about that.
Your face.
Anyways, so like I said earlier, whispering would be a weak stimulus and yelling was a loud stimulus. I'm not going to do it again or I'm going to throw it down. But so obviously both of those are strong enough stimuli to produce a sound. You can hear that I'm whispering, you can hear that I'm yelling. So giving you an example like that is really kind of hard. But there are a lot of stimuli that are strong enough for you to produce a sensation, but not exactly strong enough to reach the level of conscious perception. For instance, you're wearing clothes right now. Until I said that, you probably weren't really thinking about that. Okay. So the pressure of the clothes against your body is a sensation, but and that's strong enough sensation to activate the first sensory neuron. But once it gets up into the brain, other stimuli in there will add hyperpolarizing graded potentials and cancel out the signal, and your brain will never really process that stimulus is there. That might be a way to think about it. So at the level of conscious perception, it never gets to the level of threshold in your brain, but it is strong enough to reach threshold at the level of your skin where the stimulus is actually interacting with things. Okay. Now, this picture didn't quite go the way I'd hoped, but it does kind of suggest, whoops, wrong picture, what's going on here. It actually took me a little while to realize that there's more colors here than I thought. So down here at resting potential, that's kind of a yellowish color, right? Okay. And then green shows us a depolarizing graded potential. And then more green shows us summating on top of that, another signal. And then purple shows a hyperpolarizing current that brings it down. And then the surprise to me that I didn't realize until a student pointed out, this is actually yellow again, isn't it?
Yeah.
Yeah. What this is representing isn't a graded potential. It's just that we've depolarized and slightly hyperpolarized. So the membrane has gotten to, let's call it -60, but the signal's over. And so the membrane is going to start to move back towards resting potential. So there's no graded potential taking place during this little period here. That's just it's returning to trying to get back to resting. But then another signal comes along and we depolarize. And then another hyperpolarization, and then a depolarization. And we hit threshold and we switch from green to yellow here. This is representing the action potential. Okay. This shows kind of well the idea that we can see sort of changes in the membrane potential as different graded potentials are taking place. And eventually we can get to threshold and see an action potential occur. When I was envisioning this picture, what I was thinking about was it would look different, but I think this actually kind of gets the point across pretty well. Now, the what do you call that? Key over on the side, green is labeled EPSP, and purple is labeled IPSP. And these are actually another type of potential that I want to define. Okay. These are postsynaptic potentials. This is a graded potential at a synapse. There's EPSPs are excitatory postsynaptic potentials. And IPSPs are inhibitory postsynaptic potentials. Okay. We use the term excitatory and inhibitory because we're talking about the effect it has on creating an action potential. Okay. So excitatory postsynaptic potentials are going to get closer to threshold and excite the neuron. Inhibitory postsynaptic potentials are going to get further away from threshold and inhibit the neuron. So really what we're talking about here is EPSPs are depolarization and IPSPs are hyperpolarization. So these are just different terms for what I was just showing you. The depolarizing graded potentials are excitatory, the hyperpolarizing graded potentials are inhibitory. Okay. Now, the figure I just went to a second ago to point that out is just about graded potentials, and those are not necessarily postsynaptic potentials. They could be graded potentials anywhere based on any of those stimuli there that I've listed. Postsynaptic potentials are the graded potentials that are dependent on neurotransmitters. Okay, neurotransmitters released in the synapse, and some of them cause depolarization and some of them cause hyperpolarization. Okay, so we have what are sometimes referred to as excitatory neurotransmitters and inhibitory neurotransmitters. Now, calling neurotransmitters excitatory or inhibitory is just getting at whether they cause EPSPs or IPSPs. It makes me cringe just a little bit because the neurotransmitter binds to a receptor, and the receptor is what causes the change in the membrane potential. You can have a neurotransmitter that binds to one receptor and causes excitation, and the same neurotransmitter binding to another receptor and causing inhibition. Okay? So if you hear excitatory and inhibitory neurotransmitters, understand that it's really about the receptor for that neurotransmitter. And not all neurotransmitters would be classified that way. So, we've already talked about the synapse a little bit. The book goes into a little bit more detail, but we talked about the synapse in regard to the neuromuscular junction. So this just defines it a little bit more generally and the different things that play a role. And here's a picture of that, of a synapse, which is very similar to the picture that we saw of the neuromuscular junction. Just now we're looking at two neuronal pieces. The axon that ends in the synaptic end bulb here is called the presynaptic element, and then the target neuron is called the postsynaptic element, or actually it's labeled here as postsynaptic neuron and presynaptic neuron. But presynaptic just means the side that's releasing the neurotransmitter, and postsynaptic means the side that's receiving the neurotransmitter. And so postsynaptic membrane potentials are, postsynaptic potentials are what are happening in the target cell there. Now, there are different systems of neurotransmitters that I want to talk about a little bit, which have to do with the chemicals that are found there and the receptors that they interact with. There's two basic types of receptors. One type of receptor is what we've already met. It's an ion channel that's activated by a neurotransmitter binding to it, what I earlier called a ligand-gated ion channel. Okay. The other type, the receptor, is one protein that through a series of changes inside the cell will cause ion channels elsewhere to open up. Okay. In the book, I'm referring to this as direct activated or direct activating receptors and indirect activating receptors. Okay. So either the receptor is an ion channel that opens up, or the receptor causes ion channels elsewhere to open up. Okay. The difference between these two is a trade-off between speed and effect, or I should say speed and broadness of effect. When the neurotransmitter binds to the receptor, it opens immediately. It happens within microseconds. And so you immediately get a response. Here, when the neurotransmitter binds to the receptor, changes occur inside the cell, which then eventually spread to where the ion channel is. That can take milliseconds to happen. So these are slower than these. Direct is fast, indirect is slow. The thing is you can imagine from this picture, we can see the signal spreading to an ion channel here. If we have more than one ion channel, they'll all be activated together because this signal that's spreading here can spread to other ones around there. And so one receptor can cause a lot of ion channels to open, which might make for a stronger response. Okay? So direct action receptors are very, very quick, but only one ion channel opens when something binds to it. Indirect ones are slower, but it's possible that multiple ion channels can be opened up simultaneously, making for more ions coming in and a stronger result in the end. Okay. Now, this table here is talking about different types of systems, okay? I want to kind of separate out some information here. The first system I want to talk about is called the cholinergic system. And really, when we talk about neurotransmitter systems, we usually, the title for the system ends in ergic. Erg ERG is the root of the word energy. So it means what happens from this particular neurotransmitter. Cholinergic is referring to acetylcholine. So the choline in acetylcholine is where the choline in cholinergic comes from. This is talking about the system where acetylcholine binds to receptors and causes changes. It seems kind of strange probably to call it a system because it's based on just one neurotransmitter, but there's more than just the neurotransmitters at play. There's also receptors. Acetylcholine binds to, actually I'm gonna make this bigger, binds to a couple of types of receptors that are called nicotinic and muscarinic receptors. These receptors are named because two different drugs can be used to activate them independently. Acetylcholine will activate both types of receptors, but nicotine will only activate the nicotinic receptors, and muscarine will only activate the muscarinic receptors. Muscarine does not activate nicotinic receptors. Nicotine does not activate muscarinic receptors. So there's two types of receptors that acetylcholine can bind to. A good way to illustrate this idea is to think about keys and locks. Now, I used to, or the school used to have a different locking system for the doors that we have. And so now it's changed. I just have this one key that opens all of the doors that I need to get into. But I used to have 3 different keys. One key opened up the classroom door. Okay. I put in the lock and I turn it and I get into the door. Okay. Another key I had opened up the AV room, which is across the hall. I'd stick that in that different key in a different lock, turn it, and it opened it up. Okay. And then my third key, which I used as my office key, was actually a master key. And so I could use that to open my office, to open the classroom, and to open the AV room also. We can ignore the office thing. We're just comparing these 2 doors. Okay. Now, the master key I had fit into both locks because they're both the same brand locks. And in both locks it could turn and open it up. But the classroom key could fit into this lock and turn and open up. But if I could put it into that lock, but it wouldn't turn. Okay? And the AV room key could fit into that lock across the hall, and I could turn it open up. I could put it into this lock, but it wouldn't turn. The master key is like acetylcholine. It can open both doors. The classroom key is like nicotine. It can open the classroom door, but not the AV room door. The AV room key is like muscarine. It can open that door, but not this door. Okay? That's a way to kind of illustrate this idea. Now, the nicotinic receptor is actually the receptor we've already met, that is at the neuromuscular junction. When acetylcholine binds to that, it's a channel that opens and allows sodium into the cell. It depolarizes and causes an action potential. Muscarinic receptors we're going to meet in AMP2. There's actually a few types of muscarinic receptors. Some of them will cause depolarization, and some of them will cause hyperpolarization. So the acetylcholine down here tells you the nicotinic receptor depolarizes. Muscarinic receptors can cause both depolarization and hyperpolarization, depending on which specific muscarinic receptor it is. Okay. Now, as I'm going over this, I'm skipping over the elimination part. I don't want to get into explaining that here, given the time that we have. The next column here isn't really a system to itself. These are several different systems that are all based on amino acid neurotransmitters. So there's glutamine, glycine, and GABA. Okay. Glutamine is glutamine and glycine are amino acids that we use to make proteins in our body, but they're also used as neurotransmitters. GABA is an abbreviation for what's called gamma aminobutyric acid. I said that very quickly because I didn't want you to write it down. We only ever refer to it as GABA. Okay? It is a enzymatically modified version of glutamate. Okay? It's based on glutamate, but it's been changed. It's had some bonds broken and other things attached to it. So it's a different molecule, but it's a variation on glutamate as an amino acid. So we actually are looking at three systems here, what we call the glutamatergic system, the glycinergic system, and the oddly sounding GABAergic system. Okay. Glutamate binds to glutamate receptors, glycine binds to glycine receptors, and GABA binds to GABA receptors. Now, all three of those have pluralizations there. There are multiple types of glutamate, multiple types of glycine, and multiple types of GABA receptors. Actually, I'm not positive that there's multiple glycine, but we'll say there are. So we're actually talking about three different systems, but for each system, we're saying it has one kind of receptor. Glutamate always causes depolarization. Okay? No matter what type of glutamate receptor we're talking about, it's going to cause depolarization. Glycine and GABA, both through their separate receptors, cause hyperpolarization. Now, earlier I said sometimes you hear about excitatory or inhibitory neurotransmitters. This is really where it is. Glutamate is usually referred to as an excitatory neurotransmitter or excitatory amino acid because it always causes depolarization. But it's really the receptor that's causing the depolarization. And then glycine and GABA are referred to often as inhibitory neurotransmitters because they're always going to cause hyperpolarization. Then the next column there is a class of molecule called biogenic amines. And there are three major types of neurotransmitters in there that I want to mention. There's serotonin, dopamine, and norepinephrine /epinephrine. Serotonin belongs to what's called the serotonergic system. And it has its own receptors, which are called 5-HT receptors, because 5-HT is the abbreviation for the chemical structure of serotonin. And all 5-HT receptors cause hyperpolarization, which actually got left out at the bottom of that column there. But serotonin is strictly inhibitory, just like glycine and GABA. Okay. Glutamate is strictly excitatory. Glycine, GABA, and serotonin are strictly inhibitory. Acetylcholine can do either, and then dopamine and norepinephrine can do either also. Dopamine belongs to what we would call the dopaminergic system, and it's got two types of receptors called D1 and D2 receptors. I usually get this backwards, so I don't really care anymore at this point, as I'm talking about here. We'll pretend what I'm about to say is in fact correct. D1 receptors cause excitation and D2 receptors cause inhibition. I might have gotten that backwards, and at this point it doesn't matter. The point I'm making is one type causes excitation, the other type causes inhibition. And then norepinephrine is kind of an unusual system to talk about name-wise. First off, norepinephrine is a neurotransmitter that we find in the nervous system. Epinephrine is a related molecule that really isn't a neurotransmitter. It's really a hormone. I put it in here in parentheses because it might actually be found occasionally as a neurotransmitter here and there, but effectively we can kind of ignore it and just think about norepinephrine. Now, epinephrine and norepinephrine are Greek names for molecules that have a Latin equivalent. Okay? And obviously the similarity between the two comes from epinephrine. Okay? The nor ending, prefix on norepinephrine just is a little chemical quirk to that molecule. Epi means on top of. Nepher means kidney. Okay? Both of those are Greek. We can say the same thing in Latin. the prefix that means on top of is ad, and the root that means kidney is renal. Okay? So epinephrine in Greek is the same thing as adrenaline, which is the Latin version of that word. Okay? So you've probably heard of adrenaline. You've probably not heard of noradrenaline, but there is such a thing called noradrenaline. The reason why there's a Greek and a Latin version of these names for that molecule, those molecules, has to do with a little quirk in United States copyright law, or not copyright law, but trademark law. Back in the 60s, a drug company started producing a therapeutic version of adrenaline, which they marketed under the name adrenaline. So they trademarked the word adrenaline. When researchers wanted to study the effects of that molecule, not the drug, but the molecule that's naturally produced in our body, they wanted to write a research paper on that. They had to deal with the trademark on the word adrenaline by the drug company. So what the researchers did, instead of having to worry about getting rights from the company, they just started calling adrenaline epinephrine. Okay? And they called noradrenaline norepinephrine. In science, the term epinephrine and its the other one, norepinephrine, are used by scientists in the United States. Scientists outside the United States just call those adrenaline and noradrenaline. Now, we use adrenaline in common usage now because that's not something that can be trademarked, just like you use Band-Aid and Kleenex, those sorts of things. And you don't have to get permission to call things by those trade names. But in a publication, that's something that had to be worried about. And if the scientist was going to publish a paper that would say something that the drug company didn't like, they might say, well, you can't use that word, we own that word, and that would compromise the science. So scientists just use epinephrine and norepinephrine instead. They are collectively part of one system. So this is really the first place where we see two different molecules that are part of a system. That system is referred to as the adrenergic system. The adren coming from adrenal, as in adrenaline, and then ergic. There are two types of receptors for norepinephrine, the alpha and the beta adrenergic receptors. There are, I think, 3 alpha and 2 beta types, and some of them cause depolarization, some of them cause hyperpolarization. I can never remember. So in the biogenic amines column there, serotonin is always inhibitory. Dopamine and norepinephrine / epinephrine can do either of those two things. Okay, so acetylcholine, dopamine, norepinephrine, epinephrine, it depends on the receptor what actually happens. Glutamate, glycine, GABA, and serotonin are all specifically one or the other because all of their receptors do the same thing. The last column there, I never talk about anyways in class, which is there are proteins that act as neurotransmitters sometimes also called neuropeptides. And they do various things, which I don't have any questions about in any of the assignments, so I'm not going to bother talking about it. Let's see, where am I here? Let me just see where we are in the book. Yeah, so that was the table I was just looking at. That gets us to the end of this part of the chapter, which is the end of the chapter completely. So I did successfully get through all of the information that I wanted to. And I also did not successfully get to the rest of the neuroanatomy stuff. So the neuroanatomy lab quiz, which tech right now says that it's due before next class, I'm going to change that due date so that it's due before the class after that, because we're going to deal with that lab material, finishing up that lab quiz next week. While I'm thinking about it, this may or may not apply to any of you, but I want to be sure everybody's aware of this. Monday is Patriots Day. It's a holiday. The school is closed. Wednesday day classes follow a Monday schedule. Wednesday night classes stay the way they're supposed to be. So if you've been told that Wednesday follows a Monday schedule, that doesn't apply to our class. That only applies to any classes that start before 4 o'clock. If you happen, I don't imagine any of you have a Wednesday night class because you have Wednesday, I'm sorry, I don't suppose any of you have a Monday night class because you have a Wednesday night class, but if you do. Wednesday night classes, of course, are canceled because there's a holiday on Monday. Those meet Tuesday night next week, which may not apply to any of you, but I just want to be sure everybody's aware of this. No classes on Monday because it's a holiday. Tuesday day classes meet normally on Tuesday. Monday night classes meet on Tuesday night. Monday day classes meet on Wednesday. Wednesday night classes meet on Wednesday. And then Thursday and Friday are normal. But just so you know, as far as we're concerned, we do have class tomorrow, next week, no matter what anybody said. And we'll continue on with this stuff next time. Have a nice night.